Hello friends, we have started a new video series on the topic eukaryotic cell cycle. In my last few videos, I discussed in detail about the interphase of the cell cycle, which include three important phases, mainly G1 phase, S phase and G2 phase. And I also discussed M phase. M phase actually have three different type of strategies for division of the cell like binary fission, mitosis and amitosis. We have already discussed in detail about the binary fission or called amitosis, and now we have start discussion on mitosis. Actually mitosis process is equational division or it is a somatic cell division, and it having two different phases like karyokinesis and cytokinesis. Karyokinesis is nothing but the division of the nucleus which occur in four separate stages like prophase, metaphase, anaphase and telophase. And now in this video, I am going to mainly focus on cytokinesis. It is the process of division of the cytoplasm. Okay, so cyto means cytoplasm and kinesis is the division. It is the last stage of the mitosis in which one cell divide to form two daughter cells. Okay, the process of cytokinesis begin in the late anaphase, okay, and it complete in the telophase. Okay, that means it is not a separate stage but it run simultaneously with the anaphase and telophase. Why it is necessary to run cytokinesis or start cytokinesis during the karyokinesis? This is because any animal cell, if we consider human cell also, have very short time for completing entire mitosis. So cell have to run some stages simultaneously. Okay, if we consider human cell or animal cell, total time for the cell cycle, one cell cycle is 24 hours. That means if a human cell want to divide by mitotically, it will going to have 24 hours for one cell division. Out of this 24 hours, G1 phase of the interphase require 11 hours. Okay, it is the longest phase, as we know that many important activity is take place during the G1 phase of the cell cycle. So it is the longest and most important phase, and in this phase cell prepare itself for S phase. So G1 phase require 11 hours out of this 24 hours. Then S phase require 8 hours. We know that S phase is the phase in which DNA replication is take place inside the nucleus of the cell. Okay, so the process of DNA replication require 8 hours, and G2 phase, which is the last phase of interphase, require 4 hours. In G2 phase, cell organelle get divided, then actin filaments get produced in the form of tubulin subunits, and phosphorylation also occur in which cell produce energy in the form of ATP, and this process require 4 hours. So total 23 hours cell require for completion of the interphase only. So out of this 24 hour, maximum period is spent for interphase. Only 1 hour is remain for M phase now, and M is the mitotic phase. So cell have to complete mitosis in remaining 1 hour. So all these processes of the mitosis take place in the 1 hour. So it is a quite less time, and cell have to perform many activities simultaneously so that cytokinesis occur in collaboration with anaphase and telophase. Okay, in this video, I am going to mainly focus on type of a cytokinesis or the process of cytokinesis that take place in animal cell and plant cell. Actually, cytokinesis process in animal cell occur by cell furrow formation. If we consider cytokinesis in animal cell, it occur by furrow formation. It is the cleavage type of cell division, while cytokinesis in plant cell occur by cell plate formation. I am going to discuss both these processes in detail in this video. So first we are going to start with cytokinesis of the animal cell, which occur by cell furrow formation. Suppose this is the animal cell which have already completed karyokinesis, and now this cell is entering in cytokinesis. So after completing karyokinesis, we know that this cell will going to have cytoplasm. Okay, this is cytoplasm. Then cell will going to have two nucleus. Why two nucleus? Because as cell is coming in cytokinesis after karyokinesis. So cell already have two nucleus. Inside the nucleus you can see thread-like structures called chromatin, which is produced after decondensation of the chromosome in telophase. I have already discussed about this process. So these two nucleus will going to have chromatin network and nucleolus. Okay. At the start of the cytokinesis, this cell will going to have all the organs which is generally present in a eukaryotic cell, but in double quantity. That means if the cell will going to have mitochondria, okay, normal cell have one mitochondria, but at the start of cytokinesis, this cell will going to have two copies of mitochondria, which is nothing but the powerhouse of cell. We know that all the type of oxidative phosphorylation processes take place in the mitochondria. That means ATP generation mainly occur in the mitochondria. The cell will also going to have, say, endoplasmic reticulum, both smooth and rough endoplasmic reticulum associated with both the nucleus. Cell also going to have like other organs like Golgi complex, 2 in number, then bodies like peroxisome, lysosomes. Okay, so everything is present in the cell but in double quantity, and now this cell is ready to enter in the cytokinesis. Okay, it is a animal cell, so outer covering is cell membrane. We know that cell wall is absent in animal cell, only have outer cover which is a phospholipid bilayer, nothing but the cell membrane. This is mitochondria, endoplasmic reticulum, ER, Golgi bodies. Okay, I am not drawing everything, but just to get things clear, I have shown cytoplasm, two nucleus associated with the endoplasmic reticulum, mitochondria, Golgi bodies, okay, and cell membrane. Now, at the start of the cytokinesis, what will going to happen? A plate is produced, a ring is produced at the center of the cell, okay, and that ring which is produced at the center of cell is called contractile ring. Contractile ring. This ring is mainly composed of two different type of protein. That means this contractile ring is made up of two different type of protein. Number one, actin filament. This is the member of cytoskeleton, okay, actin filament protein. And number two, myosin 2 protein. So both these proteins are important for formation of this contractile ring at a fixed position in the animal cell during cytokinesis. But before cytokinesis, these two proteins are generally present in inactive form. At the start of cytokinesis process, another protein called Ru-A protein. Ru-A protein activate actin and myosin 2 protein, and now these proteins are responsible for this contractile ring formation in the animal cell. After formation of this contractile ring at a fixed position, now the actual process of cytokinesis is initiated in the animal cell. The next stage is contraction of the contractile ring around the cell membrane of the cell. For example, this is cell and this is the contractile ring. Now this contractile ring will going to get contracted around the cell membrane. A force is produced, a pressure is generated, on the cell membrane, and due to which, suppose this is contractile ring, first stage, second stage of cytokinesis, and due to contraction of the contractile ring, force is produced on the cell membrane which form furrow like structure. Okay, cell membrane get pinched inside, a cleavage is initiated, and this type of structure can be become visible. Okay, and due to formation of this type of structure, two compartments are produced. In both the compartment, equal distribution of material occur. That means this material get equally distributed between these two compartments. Both the compartment have nucleus with chromatin and nucleolus. Then endoplasmic reticulum, other cell organelles like mitochondria, Golgi bodies, and all other organs get equally distributed in both this compartment. And now, due to further contraction of the contractile ring in later half of the cytokinesis, cleavage occur and two daughter cells get separated from one another. This will mark the end of cytokinesis in animal cell. So now at the end of cytokinesis, we will going to have two separate daughter cells, each with two mature nucleus, contain chromatin, nucleolus, and the cell will also going to have all other cell organelles. Okay, and these are the daughter cells. In this way, cytokinesis process occur in the animal cell. Contractile ring lead to furrow formation. So, animal cell cytokinesis occur by cell furrow formations. It is called as cell furrow formation cytokinesis. Finally, due to cleavage process, daughter cells get separated from one another. The process occur very accurately so that newly formed daughter cells always going to have equal amount of genetic material in their nucleus, and all other organelles are also get equally distributed in daughter cells. Okay, this is all about cell furrow formation process occur in the animal cell. Now we are going to discuss cell plate formation process which occur in the plant cell. Suppose this is plant cell. At the start of cytokinesis, this cell will going to have double quantity of cytoplasm. Okay, cell have two nucleus with nucleolus and chromatin network. Then this cell will also going to have mitochondria, powerhouse of the cell. As it is a plant cell, the cell have a special type of organ called chloroplast. This is chloroplast, which play important role in photosynthesis process. Then plant cell also have large structure, air-filled structure called vacuoles, and all other organs are also present in this cell in double quantity like endoplasmic reticulum associated with the nucleus. This endoplasmic reticulum, which is present in both plant and animal cell, play important role in detoxification of the toxic material as well as in protein synthesis, because ribosomes are present on the endoplasmic reticulums. Okay, all other body like lysosome, peroxisomes are equally or present in the plant cell at the start of cytokinesis. Now, when this cell enter in the cytokinesis, cell plate formation process begin. What is cell plate? It is nothing but the cell wall like structure which is produced at a fixed position in the cell. Actually, this cell plate in the plant cell is made up of vesicles coming from Golgi complex. Okay, suppose the plant cell have Golgi apparatus or Golgi bodies organ which is also present in animal cell, and we know that the main function of the Golgi apparatus is delivering protein inside the vesicle to different organs. So this Golgi complex produce vesicles, and now these vesicles contain cell wall component. Okay, the vesicle which produced by the Golgi complex contain component of the cell wall, and now this vesicle transfer to the center of the cell where they produce, suppose this is vesicle, where they produce cell plate. So this cell plate is nothing but the structure containing component of the cell wall. Okay, component required for synthesis of the cell wall coming from Golgi complex. Okay, we can write first stage of the plant cell plate formation, which is generally occur due to delivering cell wall component by Golgi bodies. This structure which is formed at the center of plant cell due to these vesicles is initially called phragmoplast. And this phragmoplast later on going to convert into cell plate. Okay, now in the next stage of cytokinesis in plant cell, this phragmoplast increase in size at the center of cell, which will again form two compartments similar to the animal cell in plant cells. Okay, this phragmoplast or cell plate going to divide cytoplasm and all other component of the like nucleus, all other component of the plant cell into two equal parts. Okay, if you want, you can write name of every component like nucleus, liquid portion cytoplasm, mitochondria, endoplasmic reticulum, ER, vacuole, okay, lysosome, etc. And at the end of cytokinesis in plant cell, due to increase the length of plate at the center, a new cell wall is produced at the center of the plant cell, which finally divide two cell. So at the end, two small size daughter cell having cytoplasm, nucleus get separated. Both the cell will going to receive equal quantity of cytoplasm and one copy of every cell organs, including mitochondria, chloroplast, and all other organs. So this will be the end of cell plate formation cytokinesis in plant cells. These are daughter cells. This is all about cytokinesis process occur in animal and plant cells. In this way, we finished the second type of cell division, that is M phase, which is mitosis. In last few videos, I discussed in detail about the karyokinesis of mitosis, which complete in 4 phases, mainly prophase, metaphase, anaphase and telophase. And in this video I discuss about cytokinesis of the mitosis, which complete in 2 phases, that is cell furrow formation in animal cell and cell plate formation in plant cell. From my next video I am going to start with last type of cell division, that is meiosis. Thank you.